The silverleaf whitefly, or the Baissima tabacai biotype B, is now widely known as Middle East Asia Minor 1 species, or MEAM1. It was first identified as a new strain of B. tabacai when it appeared in North America during the 1980s. Vast numbers of these whiteflies were found to be infesting winter vegetable crops, causing an estimated $500 million loss in the winter harvest in 1991. Investigations of the new species led to the discovery of its multiple unique features. The biotype B strain differed from its indigenous counterpart, the American strain, or the A biotype, by the ability of its larvae to induce a phytotoxic silver leaf disorder in squash. The biological traits of MEAM1 implied that it had evolved in an intensive agricultural region with high exposure to pesticides. This environment allowed the new species to feed on a wide range of crops and develop resistance to pesticides. In addition to its newly found genetic superiority, MEAM1 also showed a second dangerous characteristic. Like the common mosquito, the biotype B has the ability to act as a vector, or a delivery system, for many plant viruses. The biotype B species is known to host as many as 60 different plant viruses. Some viruses this species has been known to carry include the tomato yellow leaf curl virus and the tobacco curly shoot virus. The indirect mutualism between the B biotype whitefly and these viruses may contribute to the ability of the B whitefly biotype to effectively invade crops, displace indigenous whiteflies, and spread disease caused by the various viruses, as compared to the indigenous biotype A, which has no such mutualism.
Due to time constriction, we will go over a quick crash course of the methods used in this experiment. For each biotype, approximately 300 newly emerged adult whiteflies were collected from the culture on cotton and divided randomly into 3 groups of around 100 and more, and these groups were inoculated onto 3 types of tobacco plants: healthy, curly shoot infected, and yellow leaf infected. On each type of plant, approximately 30 replicates were conducted. The flies were placed onto their allotted plants when the plant reached its 5-leaf life stage. Once the plant was at its 5-leaf life stage and continued growing till it reached its 7-leaf life stage, the experiment was ended and data was collected. The data that was collected for these experiments were mean egg count, longevity of those eggs, mean development time, mean number of nymphs, which are juvenile flies, and lastly, the mean number of adults.
In the first experiment, the scientists compared the indigenous ZHJ1 and the invasive B biotype whitefly on healthy and the 2 infected tobacco plants. The whiteflies were first grown on cotton, which is a non-host for the begomoviruses, and transferred onto the 3 plant types during their adult stage. Taking a look at Table 1, we can see that the B biotype had a higher fecundity and longevity than that of the ZHJ1 biotype when incorporated with infected plant. In comparison, the ZHJ1 biotype did not have much of a difference between the 3 plant types, suggesting a lack of mutualistic relationship. The next experiment was similar to that of the first. The only difference was that the whiteflies were placed on the plant upon emergence from their egg. The main thing to take away from Table 2 is that there was an increase in both the mean number of eggs laid per female and the mean longevity for the B biotype living on infected plants compared to those living on healthy plants. In comparison, ZHJ1 did not seem to have much of a change in either variable, but had a low N value. The next experiment dealt with comparison of the population increase of the B biotype whitefly on the 3 different types of tobacco plants. The scientists found that the mean number of infected plants were 2.1 and 12.8 times higher than that of the healthy plants for all life stages. Finally, scientists wanted to test the relationship of the vector and the virus in the absence of the host plant. Therefore, they examined 3 types of whiteflies: non-viruliferous and the 2 viruliferous whiteflies. The performance when placed on the cotton plant were recorded. It turned out that the B biotypes with the TBCSV infection had an increase in fecundity, while the TYLCCNV infected ones had a decrease in both fecundity and longevity. This supports the idea of indirect mutualism, in that there needs to be a host plant for the mutualism of the vector to whitefly to occur.
So diving straight into the analysis of Table 1, if we look at the mean number of eggs found in biotype B, we can see that there's an overwhelmingly larger number of eggs found than that of the ZHJ1 biotype. Another thing we can notice from this figure is that the mean longevity of biotype B is also much, much larger. These two components added together causes a much larger benefit for biotype B, giving it a much bigger advantage. Lastly, we can look at the comparison between the healthy plants and that of the infected plants. The data for biotype B shows a strong correlation between the infected plant and the amount of eggs that were produced on said plant. The same cannot be said for ZHJ1, however. Its dataset shows that when the whiteflies were on the infected plant, there really wasn't any difference in the amount of eggs that were laid, and no difference to longevity as well. Table 2 shows the performance of the two different biotypes on the three different plants that were used. If you look at the table, you'll notice that the right half of the table has very small n values. This was due to experimental error, and because of this error, the mean number of eggs and the mean longevity that was calculated isn't entirely accurate, so I won't be talking about those particular data sets. However, the percent survival and the mean development time are applicable. So if we look at the B biotype for percent survival, we can see a very, very large correlation between the number of flies that are surviving and the number of flies in the ZHJ1 dataset that are surviving. It's almost 500% times that of the ZHJ1, and the mean development time is larger as well. As I said in Table 2, with the mean number of eggs and the mean longevity being higher in biotype B, this gives the B biotype a much, much greater advantage over the ZHJ1. Looking at Figure 3, we can see 4 graphs showing the mean total of eggs, nymphs, adults, and the total of all of those respectively. What you want to notice about this figure is that all the trends in all 4 graphs are very similar, and all show that the yellow leaf infected plant does surprisingly well as far as mean numbers goes across the board. The plant that did the worst as far as number of flies was the healthy plant, which is great evidence behind the symbiotic relationship between the disease and the fly that is present. Table 4 shows the fecundity and longevity of non-viral and viral females for the two biotypes, as well as the survival and development time of the progeny produced by the females on healthy cotton. So if you'll recall back to the last figure where there was the 4 graphs all showing that the largest mean number of eggs, nymphs, and adults was found on the yellow leaf infected plant. However, if we look at this data set, we can see that for both biotypes B and the ZHJ1, we can see that the yellow leaf infected flies actually did the least well in regards to mean number of eggs and longevity than the other two groups. Because of this data, it would lead me to believe that the yellow leaf infection actually hinders the fitness of the female fly and positively affects that of the male fly. Another thing to point out about this figure is the mean development time. It can be seen that the B biotype actually has an overall longer development time than that found in the ZHJ1. However, because of the results found on the number of eggs of biotype B compared to that of ZHJ1, it can be concluded that the difference in developmental time is not significant enough to give ZHJ1 a benefit over the B biotype.